So, hello everyone. This is Simrat Kaur, and my friend Manroop is here with me.
Hi,
So, we
hi
are
guys.
students of Capilano University. Today, we are doing She is Manroop, and today we are doing a group presentation on the rate of production of carbon dioxide from yeast fermentation by using different kind of sugars. So, in this experiment, we are going to see how much is the different rate of carbon dioxide when we use different kinds of sugars. So, over to my friend Manroop.
Hi guys, hi everyone. So
Hey
this is
Manroop.
Manru. I'm just gonna share my screen and then I'll just present it with you. So let's just start it. So here the topic of our research is the effects of sugars on the rate of production of carbon dioxide by fermentation of yeast. Before telling about the research, I just want to go over through about it, just about the introduction. Like we all are familiar that yeast are single-celled organisms. The species, they are also known as Saccharomyces cerevisiae, which is also called baker's or bear's yeast. Yeast, which are facultative aerobes, and it leads to the formation of ATP as well as by the process of oxidative phosphorylation. So, we already know that fermentation is an anaerobic process. So we are just going to taking it place by using fermentation of yeast. And let's move towards our second slide, which is showing about the effect, like the introduction. Like the question about our research is, what is the effect of different sugars on the rate of anaerobic respiration of yeast by the amount of carbon dioxide produced? Before proceeding it further, I just want to ask like a question about this chemical reaction, like how glucose reacts with oxygen. And my friend Simmer is just going to explain a bit of this reaction. Simmer, over to you.
So, hi friends, in this is the reaction you see. In this, glucose is oxidized, and it produces the six molecule of oxygen with some amount of carbon dioxide, water, and here ATP is the energy released in this reaction. So, here is the energy released. Glucose is so yes, the thing is glucose is used to make the ATP. The work is the chemicals. So yes, over to Manroof.
Oh, yeah, there was a bit connectivity problem maybe. I'll just explain about the methods then. Like we are familiar that we're gonna use glucose, maltose, sucrose as our three different sugars that we are gonna use in order to test the effect on the production of carbon dioxide using yeast fermentation. So here there are control group controlled variables as well as some independent and dependent variables which are being used up for the method. So I just want to ask, like, what is control variable? What are different kinds of dependent and independent variables? So Simmer is going to explain a bit, like defining it for you. So Simmer, over to you.
Yes. So friends, here we have three kind of variables: independent, dependent, and control variable. So first of all, independent variable. Independent variable is the one thing that we can change, limit to one in an experiment. So then the second one is dependent variable. The change that happen because of independent variable. So dependent variable is that change which is occurring due to independent variable. So the third one is control variable. So here everything we want to remain constant or that we don't want to change is considered as control group. So to explain it further, I want to take help from an example. So let's say we have a plant in a pot. So the water which we use to the liquid we use to water each plant is called independent variable that we cannot change the liquid, that is the water. So the height of plant is dependent because this is change occurring due to independent variable. The height is increasing due to water, so water is independent and height is dependent. And the third one, that is controlled group. So here controlled is the amount, the other things like the amount of liquid, soil type, and other that things. I hope it works.
So
Over to Manroop.
yeah, thank you, Simrat. Okay, she just explained about the definitions of this, and I'm just going to move further from the methods to results and interpretations. Firstly, I just want to like discuss about the research question that we just were having, like the effects of different sugars, and why baker's yeast is used because it grows very quickly and efficiently in anaerobic conditions. So that's why the baker yeast has been used up for the production of CO2, and the different sugars which we are going to use it, they have some different impact on on the on it. So let's move further towards the results. Firstly, I just want to say you that this is a physiological mechanism. So this is not an environmental, this is a physiological mechanism in which we have looked at the uses of glucose, fructose, and maltose, like how they have affected the change in the volume. And we use standard deviation method and mean progression, that is the CI method that we just use, where we just kept the constant, as well as the source of error is also kept at 8. So
So just a question, Manroop.
yeah,
Can you please explain a bit what is N? Like what is the 8? N8.
that means that the error is constant over there, like it's been kept same.
Oh,
Yeah,
oh, okay. Yeah, I got it. We performed the experiment for 8 times.
yeah.
So this is and like the number of time we are performing that experiment.
Oh yeah, yeah, you're right. That is for the
Mm.
number of times we are performing the experiment. Okay, so let's move towards the graph, like the figure which we have done in order to explain how glucose has impacted the change in the volume after fermentation, as well as maltose and sucrose. Firstly, we can just look in the figure that glucose has really changed the volume by more than 9, as well as sucrose. But you can see on the x-axis that maltose just change a bit lower. The change in volume is less. So we can analyze from this figure that maltose has little effect as compared to glucose and sucrose. So the production of CO2 is less. So the production of CO2 is less in maltose and more in glucose as well as sucrose. So let's move towards the references. So here you can see that we have just mentioned the references, and from here we just gonna bind it up. So Simmer, can you just bind it up for us? Yeah.
Yes. So I just want to explain a bit more about why does sucrose produce more carbon dioxide as compared to glucose. So here the reason is there is an enzyme called invertase that catalyzes the hydrolysis of sucrose into both fructose and glucose, which will explain us why sucrose produce more carbon dioxide than maltose.
Okay. Okay,
Yes.
so we are
And
gonna, yeah, we are gonna end the session here. So thank you so much.
so thank you so much.